Yeah, very interesting. So next and finally, Jose Pedro Gil from Karolinska Institute in Sweden and also from Gulbenkian Institute. He will talk about clinical trials involving children in Africa. Thank you, Jose Pedro.
Thank you, and good morning. I hope you still have the courage to listen to me. I have just arrived from Angola, from a place where I've been for 1 month, not me, but touching extreme poverty during a clinical trial that we're preparing there. So it's always, when I am like 1 month or more in these places, when I come back to Europe and I come to places beautiful like this, it always takes, a couple of days to adapt myself to our wonderful reality that many times we don't realize. No, this is just for my guidance. Good. Well, experiences, says Adrian. So I prepared something. I'm going to make a public confession. I started to make this presentation yesterday, 2:30 in the morning, because I had no time on the meantime. So I'm not even sure this is the last version. Sorry about that, but I hope to be coherent. So here we are. And it all starts here, my little story. This is Zanzibar. It's quite beautiful. Quite a nice place to make clinical trials. Nowadays, not good. Not malaria left, not much. But when we started working there in 2002, it was a lot, 40-50% of incidence of malaria. And at that point, the Zanzibar government was changing for a new artemisinin combination therapy, ACT, in particular artesunate-amodiaquine. Now, amodiaquine is a very interesting drug. It works as most antimalarial drugs in the intra-red blood cell cycle of the parasite. It's metabolized by, an old friend, CYP2C8, a liver enzyme, which is quite polymorphic, not as much as CYP2D6, my goodness, but still with some interesting SNPs, especially star 2, star 3, which have interesting also geographic, distribution. Star 2, which is an enzyme that still works reasonably, it's very common in Africa. Star 3, which supposedly, it's a bit contentious, but anyway, supposedly an enzyme that has a decrease of 80-90% of its enzymatic capacity, it's very common among Caucasians, Europeans, Caucasians in general. They adopted this, and at that point, we made some phase 3B studies of artesunate-amodiaquine just before the adoption, just before the implementation of the combination in Zanzibar. I have to guide myself from here. And these are very common kind and not very complicated, in design, operationally a bit complicated efficacy studies. We just passively, the patients come to us, we treat them, and we follow them up for a follow-up with a size that is usually proportional to the half-life of the drug. In the case of amodiaquine, which is fastly metabolized to diethylamodiaquine, which has a long half-life. Typically, it's 42 days. Well, some groups make it 35. We typically make 42 days. Now, the parasite is also an actor here. And amodiaquine, supposedly the parasite can resist to amodiaquine at least with one tool, with PFMDR1. PFMDR1 is a gene that codes for a protein that is the equivalent in the parasite to our very well-known P-glycoprotein, known also from drug resistance in cancer. And in particular, this enzyme, meaning this protein, have two mutations that at positions 86 and 1246, when these two mutations are there, supposedly the parasite is less sensitive to amodiaquine. And we somehow prove that in practice by showing that when we see the frequencies of these two mutations before the treatment, when we compare with all the infections that happen after the treatment during follow-up, there's a clear selection of these mutations. So parasites with this YY haplotype they resist better to amodiaquine. So far, so good. But, my background is in pharmacogenetics long time ago in my PhD. So when I came to malaria, I couldn't just give up on my old love. So I asked myself, are there what is called hot populations? Are there humans in the middle of our patients that because of their genetic background, they facilitate resistance? And the idea was a bit like, okay, there's star 2 and star 3. If they change, apparently, the pharmacokinetics of the drug, they change the exposure of the parasite to the drug, to amodiaquine and DIAC. So, it might have some influence. So I decided to genotype all these patients for CYP2C8, especially for star 2 and star 3. And you know what? Yeah, something happened. The people that have star 3 are much more frequent to have infections. Why? Why? Well, the resistant one. It seems that not being so much able to make this transfer from amodiaquine to DIAC makes the parasite more comfortable, especially selecting these resistant ones. We published this. It was quite a curious observation. And in the meantime, we expanded more the study with another trial for another question. Does it have any clinical importance? And well, lately we have seen this. If you are a star 3 carrier, you are in more risk of having during the follow-up a recurrent infection, be it a reinfection or recrudescence, I'm not going to enter in that, a breakthrough infection after the treatment. Now, that is interesting and gives a purpose for our thing. So, this is the first story I'm going to tell. Well, I have to say that this is the framework where we live nowadays. So, what is the idea? So, amodiaquine is degraded to DIAC, but if the star 3 is there, there's more amodiaquine staying for more time in our body. So, amodiaquine tends to create these reactive compounds that probably are related with mild adverse events and very rarely with quite serious adverse events. But both of these events, one gives a very bad publicity, the very rare ones, and the mild ones, which is typically belly pain and all that, are not very comfortable. So these probably both contribute for less compliance of the patient to make the treatment until the end. And that, of course, supports the development of resistance, which is composed more also by the selection of more resistant parasites. That's the mental framework we have. I do not have a proof of all this, but that's how we work nowadays. That's the importance of pharmacogenetics here. Okay, CYP2C8 is interesting. Move on to something else. This is a strategy that is supported by WHO, and if you want to know more about it, Professor Nick White is there, and is probably much more proficient than me on this area. And the idea is relatively simple. In some areas of Africa, especially Africa, where the season of malaria is very concentrated because of the rain in certain points of the year, maybe it's a good idea, if it's only 1 or 2 months when malaria really hits, to give some prophylactic treatment to the kids, especially the children which are more exposed, to be able to pass this risk time of the year, this risk season of malaria, without, under protection. So this is typically a mass drug administration system. And of course, they need long-withstanding, long half-life drugs. And sulfadoxine pyrimethamine, they stay for a long time. And amodiaquine, not amodiaquine itself, but DIAC stays for a long time there. But it needs to be metabolized to DIAC, otherwise it doesn't work like that. So what is the strategy in the end? The idea, and what's this? Well, I don't know if it works, but anyway, I can explain that. So the areas of intervention are areas which have unstable malaria, have stable, very seasonal malaria, or drier areas, which are concentrated in the Sahel area and a bit in the south of Angola and Namibia, etc. For the pharmacogenetics point of view, it's particularly interesting the Sahel area, because north of it is more of a Caucasian area, lots of star 3s. You can see the little map there. Portugal has almost 20% of the allele frequency. In the south, when the tropical, let's call it tropical Africa, star 2 is the main one. But what happens in those in-between areas? Is star 3 frequent there, the places exactly where this new strategy, this new chemotherapy is being applied? Well, actually, we don't know. There has not been studies on that. In fact, if you go to 1,000 genomes, you realize that the African population is bloody ridiculous. You have something like Nigeria, the frequency of genes in Nigeria, they fetch 40 persons, and they say this is a sample of a population of 60 million. So these studies I think are good for grants, but they're not a big help. And there's nothing, absolutely nothing, as far as I know, from these regions which are an interface between the Caucasian and the more, let's call it Black Africa, with not actually an old term. So, what happens there? This is Eritrea. Some time ago, there are a lot of Eritreans, immigrants living in Sweden. And the daughter of one of these immigrants, which is a brilliant student of pharmacy in the University of Uppsala, came to me to study, the population of Eritrea concerning CYP2C8. It was her interest. I found it super interesting, and we made a collaboration, and the proper one, in the sense that we really took samples all over the country, because the country is ethnically quite complex. It's 380, a reasonable number, and the answer is, yeah, they are a bit in the middle. You can see here that in the African population, star 3 is almost inexistent. Zanzibar is a little bit of a special place. That's why we have star 3s, because there's a bit of an Arabian historical influence in the island. In the Caucasian ones, you can see star 2 almost doesn't exist, star 3 is the big one, and these populations are actually intermediate. And that, I think, is interesting. It's interesting also from a policy point of view, from a seasonal chemotherapy point of view, among others. There's other issues in this story, but I'm not going to enter that. We don't have time for it. And why am I, showing all this to you? I don't know if it's very interesting or not. It's for one reason. When Adrian invited me for this meeting, in the beginning I must say that I didn't completely understand I didn't grab exactly what was the idea. I just grabbed it some days ago. And the idea is actually the experience there. And the first thing I'd like to say, and from now on my presentation is, I have actually something here to keep me minimally disciplined, because I tend to talk a lot. The idea is, what can I, from my experience, contribute for the objectives of this meeting? It's not exactly this. I hope that we will publish this, so it will be free. So first of all, the pharmacogenetic studies, I think that they should be purposeful. Just going to a country and making a lot of genotyping without an idea, I see people doing that. I think it's absurd. In Africa, it's particularly absurd because the populations are so heterogeneous that in the end of the day, those papers, unless they are 60 papers in different places, are relatively useless in my view. So there are some big patterns, CYP2C8 for example, but if you want to go deep, you need bigger numbers. That's the first thing. The second point that I'd like to speak is, and I must say, what I'm going to say is not published, it's not actually, this is anecdotal. This concerns my experience, the experience of colleagues of mine, but it's things to think about. It's things from the field, from really interacting with the patients. So sense of purpose is important because sometimes the patient really, even in the poorest places, they realize when it's important to do something or not. And I have a photo. I have a photo here, is my last slide, and I will continue talking on that. I have a photo of a person. I usually don't take photos of people in Africa. Some colleagues of mine, they just take. I just don't take. I feel bad. I don't like it. I really don't like it. And I think that I wouldn't like to be photographed when I am with my kid going to some trial and the kid is sick. I don't like it. I had some ferocious discussions with colleagues of mine about it. But this one I took. There was something that I spoke a bit with his mother, And I asked her, so disclaimer, she allowed me and explained exactly that I would use it publicly, and she was okay with that. And both kids had malaria. And one thing was interesting is that when I asked that, and she told me, just do not expect me to smile. She had no reasons to smile. So I said, yeah, you do whatever you want. So, and this photo is very dear for me because it's a bit of reality that we get there. Very poor people, not trained. Some they don't know how to read and write. In Angola, it's even tougher from that point of view. And you have to explain to these people why you want blood from their kids. And that can be a bit complicated. We try, in informed consent moment, to explain why we do this. Now, for malaria, it's relatively easy because, you can, unfortunately, Africa has been in wars many times, war, enemies, these kind of concepts. They don't need American movies for that. They understand that from their life. So easy to explain to a mother that the parasite, that sometimes they don't even understand it is a parasite, is something that invades, it's the enemy. And we have the weapons, and the enemy can become resistant. They understand that point of view. So it's easy to explain why it is beneficial directly for her kid, because she doesn't care about science, she cares about the kid being treated, period. And so it's relatively easy. Now, explaining pharmacogenetics, that is something that can be important because maybe there is the risk of some adverse events. Adverse what? It's already something difficult to talk about. After all, there's risk for that. There's enough risk for you to be telling me this and wanting blood from my kid. It can get more complicated. I think when you design trials of pharmacogenetics, purely pharmacogenetics, this explanation has to be very, very well thought. And I hope that one day there will be guidelines for that, because it's a bit complicated to deal with. That's the first thing. The second thing, it's pharmacogenetics. Let's see it. Before malaria, that's the enemy. It's the parasite. Easy. It's its fault, When you start saying, well, it's your genes, it's you that might not make the drug work. It's your genes that might make your kid not feel good with this drug. Now, this is complicated because you transfer a bit the onus of, responsibility on the disease to the patient. Okay, for us in this room, this is ridiculous, but it's not ridiculous in the middle of the field. Again, it's something that has to be very well thought, and we have made many mistakes with that, in fact. The other thing is that, Adrian just told me to speak about your experience or others, so I keep on saying these little stories. Some people come from traditional healers. Some things, well, we can say, okay, so some phytomedicine, traditional and all that. We cannot actually just say, oh, come on, No, after all, artemisinin, a drug that I'm a big fan, it came from, a tea. So sometimes they have something in the pocket. And when they don't have, like all these bad spirits or something, it's important not to, we dismiss scientifically, but not to dismiss socially that. You have to, be patient with the person, understand the point of view, and understand for that person, that thing is as valuable as any of our pharmacokinetic genetic studies. And again, people, especially young students that are participating in these kind of, trials, they have to be very well explained that they have to be patient, and it's not patient, yes, tell me about it. No, they have to at least apparently give the same importance to our intervention to another one that probably didn't work, but we're not going to just dismiss like that guy in your village is a completely absurd. It's not a good thing. It's not a good thing. It's not the way to go, at least in my point. And in the end, what I mean with all this, I've been complaining a lot and not giving much solutions, but the solution for me has two parameters. First, finding the moment. The moment I come to realize is not in a moment with the doctor. The mother just wants the drug. The moment is the informed consent. That is the moment, that is the point that usually people just put the paper in front, they say, this is good for your kid, come on, sign, we need you for our trial. That's the moment where we have to interact, that's the opportunity to try to interact with the patient there. And it pays in the long term, because if people feel respected at that point, if we don't dismiss them, the next trial, they will be volunteering much easier. And so that is the point. Now, do I know how to do these things? I'm a molecular biologist, and I'm surrounded by doctors and molecular biologists and people. No, we don't. We have to engage social sciences people. I think that it's fundamental that when we design a trial, when we make the power calculations, all the logistics, the molecular work that we like so much and all this stuff, we should, and almost nobody does, have a module of social sciences to help us. This is not, how may I say, intellectual snobbishness, to help us actually on how to interact, how to optimize the study, how really to, in a way, respect the patient at that point. Because what we sometimes think is respect in Europe, it's not so respectful in some cultures, and we have had some issues with that. And with that said, I had a lot more to speak. As I said, my wife complains she's Swedish. She complains that I talk a lot. I am finishing here. I hope I'm inside the 20 minutes. Thank you very much.